Yeah,
So
sure.
sorry for taking your time. I'm overextending my stay, but
No worries at all.
Yeah, just a quick question. First of all, before that, a phenomenal work and presentation. I really enjoyed it.
thank you so much.
So I think in slide 3, I think you mentioned the Schrodinger's equations. You had H hat psi is equal to E for psi, right? So is it safe to say that H hat is equal to E, or is there any other like catches to it?
That's a wonderful question. I think in the previous, I think like our first semester, Ms. Jahanikia had asked that same question. So the thing is, this isn't an algebraic equation. These are actually matrices. So when you actually multiply them together, you get different results on either side. You only get very specific results. So you can't cancel out the wave functions because these are actually big matrices. When you multiply them, they have vastly different results. And these are also differential equations. So you can't really cancel out the size because the equation works slightly differently compared to algebraic equations. But yeah, exactly.
I see. Thanks for that. And
Of course.
just another quick like working in quantum computing and seeing that your work is very connected to quantum mechanics, I was wondering like maybe in the future if it's possible to use quantum computing to do such calculations that might reduce your runtime, increase your accuracy. Just a suggestion. That's it.
I was
Thank you so
definitely
much.
thinking about I was thinking about that as well. I would love to collaborate on that. That'd be wonderful. Thank you so much.
Yep.
Okay. There's no more questions. I think, yeah, Abby, you ready?
Yeah.
Amazing.
So my group is the Benson group, and we're investigating the efficiency of the saltwater mussel Mytilus californianus' ability to filter microplastics from aquatic ecosystems. As plastic pollution escalates, microplastics are becoming a larger threat to marine ecosystems. Plastics smaller than 5 millimeters in length, known as microplastics, largely originate from plastics degraded over time by erosion, sunlight, and other things. The most abundant type of microplastics in the ocean are synthetic polymers. Most microplastics come from the fibers of synthetic fabrics or particles from tires. Runoff, storms, and wind carry the microplastics into aquatic habitats. Microplastics impact many species' growth rates, stress levels, and mortality rates, affecting the dynamics of their ecosystems. There are approximately 24.4 trillion microplastics in the ocean, showing how massive this problem is. Mussels are filter feeders, meaning they take in water from one side and expel it out the other. There are filters in between both of their sides, which allows them to extract their food of choice, phytoplankton, from water. The filters are kind of like gills, as both filters and gills suck and pump the water through. One mussel can filter almost 80 gallons of water just to intake their daily required amount of food. Even though mussels are trying to catch food, they also catch pollution and microplastics. Mussels have the potential to filter and retain microplastic particles. Mussels continually filter out the seawater surrounding them, including microplastics floating in their area. Mussels are a potential solution that can lessen the effects of microplastics on marine ecosystems. We thought it would be interesting if mussels were efficient to work in a real-life environment. Our research is aimed at investigating the efficiency that mussels may have in filtering and retaining microplastics in hopes of addressing marine microplastic pollution. Given the increasing trends in ocean microplastic concentrations, our hypothesis was that as the size of the mussels increases and the amount of microplastics mussels are exposed to increases, the amount of microplastics the mussels are able to filter and retain will increase. It is known that mussels can intake microplastics, but we were trying to find out if they are efficient enough. Our group collected California mussels from Mavericks Beach tide pools in Half Moon Bay, California, and transported them to the laboratory. We constructed 3 tanks with conditions of 35 ppt salinity and room temperature, along with the addition of 0.1 milliliters water conditioner for every 1 gallon of water. Microplastics were placed in different volumes for each tank. 0 grams for the control tank, 0.002 grams for the low tank, and 0.004 grams for the high tank. Using an orbit sander, microplastics were created by sanding plastics types 1 to 5. The microplastics we created were routinely added whenever water changes occur. The tanks were routinely maintained with water changes, removing dead mussels and cleaning the tanks. We used mesh to prevent microplastics from being filtered out. We remove mussel tissue using a scalpel tool and later digest it using a 10% potassium hydroxide solution. We create digestions from each tank once a week. After we make digestions, they're incubated at 37 degrees Celsius for 2 days. Then we purified the plastics using filtration and quantified the number of microplastics by counting and then analyzing what plastics are present. Currently, we are experimenting with new methods to improve filtration efficiency with our digestions. One method we have tried is centrifuging the digestions for 3 minutes at 3,500 RPM. In addition, we have tested using a vacuum filtration with a 1.60 micron or 100 micron filter paper. We've also tried using various salt solutions for density separations. However, all of those had some kind of problem, so we moved on. Right now, we are particularly exploring the use of a hypersaline solution to improve the vacuum filtration process by keeping microplastics on the surface. A hypersaline solution is created by boiling 120 ml of salt in 25 ml of water. After the solution is filtered, the filter paper holding the microplastics is dried overnight. There is no universal method to quantify microplastics out of the digestions. Then the microplastics are quantified visually by counting each microplastics individually with a dissecting microscope and ultraviolet light. These microscopes are analyzed and identified using FTIR spectroscopy by comparing the FTIR baselines for common plastics to those in the microplastic samples from the digestion mussels. The findings obtained from the research will be utilized to compare the amount of microplastics filtered and retained by mussels of different sizes with regard to their varying exposure to microplastics. We have recently come to the conclusion that the hypersaline solution makes it too hard to identify the amount of plastics on the filter. We have moved on to a sonification method that we have a lot of confidence in. Instead of having the plastics on the filter like the hypersaline solution, the sonification method would remove the particles directly from the filter, so we can look at it much clearer. We do not have any quantified results because we are still in our trial phase. At the moment, we have figured out how to make digestions and put microplastics in the tank and multiple filtration ways. Our goal is to have a 90% efficiency, and when we achieve this, we can go into further testing. We are tracking the efficiency of our separation by comparing the number of plastics put in versus the amount that comes out. To find the efficiency of the mussels, we counted the number of microplastics under ultraviolet light in a dissecting microscope. We are currently testing out washing off plastics off the filter with nitric acid, which we have much hope for. Some problems that occurred during the last 2 semesters were lots of mussels dying and tanks not being maintained. This semester, we fixed both those problems by getting new mussels from Maverick Beach tide pools in Half Moon Bay and maintaining the tanks better. We cleaned algae from the tanks and did water changes more frequently. Something that looked promising at first was using a centrifuge to separate the microplastics out of the digestions. However, that did not work because it was too hard to separate and the plastics were not visible. We also attempted using density and gravity filtration. It was not separating in the way that it was supposed to because of density differences, leading to us giving up on this method. When we used a hypersaline solution, the microplastics were hard to see in the filter, which led to us not being able to get proper data and identify how many plastics were left. We are currently attempting to use FTIR spectrometry in order to quantify the number of microplastics being filtered by the mussels. The FTIR technology is also being used to identify the kinds of microplastics that the mussels are intaking, as well as the kind of microplastics that the mussels are not intaking. But our data and observations are not yet finished, and we are continuing to work on this quantification and identification. Regarding the current filtration method, it is still very trial and error. We have run into issues such as the plastics not being visible after being filtered and sediments being mixed with the filtered plastics. We hope to find a medium in the future where only the plastics float up during density separations and not other organics. In addition, the sander that we use to make microplastics is slow, but we hope to speed that up by using a coffee grinder to make the microplastics instead. We will also look up the samples we have taken every week in the future and be able to quantify how much plastics each mussel is able to contain and relate them to the size of the mussel in the tank that was put in. Although there has been a lot of trial and error separating the microplastics from the mussels, we found a few methods that could work in the future with a few modifications. The filtration process wasn't promising at first due to the microplastics flowing through the filter paper made of larger microns. Changing the filter paper to a finer size slightly fixed the issue. Since the spaces in the filter paper became smaller, the microplastics were stuck inside, and using a microscope to quantify the microplastics wasn't working out. The next method we came across to quantify was using a black light, in which the microplastics were easier to differentiate since they were throwing off a fluorescent color. We then used a centrifuge to further separate the microplastics from the mussel digestion. Because the microplastics were hard to pipette off the top, the method is not very efficient. We are working on a better way to separate the plastics. This will probably be the nitric acid method that we're currently working on right now. Additionally, we are currently attempting to use FTIR spectrometry in order to quantify the number of microplastics being filtered by the mussels. The FTIR technology is also being used to identify the kinds of microplastics that the mussels are intaking, as well as the kinds of microplastics that the mussels are not intaking. However, our data and observations are not yet finished, and we're continuing the work on this quantification identification. A huge thank you to my group and my advisor, Andrew Benson, for help on this project, as well as ASDRP and its sponsors. Are there any questions?
Sure. I'd like to start with some questions about the microplastics themselves. Are the mussels themselves harmed by the microplastics that they ingest?
It's not really known right now, but yeah, they most probably aren't harmed by them.
And do mussels have predators?
Mussels definitely have predators, but
And are the predators harmed by eating the mussels containing the microplastics?
mussels, like they get all kinds of pollution, so no matter if we do this or not, they're still gonna get polluted.
Okay, and finally, what happens to the microplastics when the mussel dies? Do they just wind
Yeah,
up
I
on
think
the seabed, or what?
They're expelled from the from the mussel.
Okay, and so let me go back now. What is the purpose of removing the microplastics from the environment where the mussel lives?
Microplastics are a huge problem to aquatic environments, and the problems that they can cause are not all known yet, but they're like really bad for the environment.
So I think it would be useful to be able to point out just to some of the specific harms that they could create.
Yeah,
I can talk on that point, and Avi will talk about that later too. But so they can do a lot of different things to anything in an aquatic environment and in a terrestrial environment as well. In smaller organisms, they can block up kind of their digestive tract or passageways, and harm them physically by hindering any sort of bodily processes, as well as some plastics can kind of be toxic for things, and so it can affect anything that ingests its like toxicity levels, which can lead to a ton of problems. Those are a few things.
Okay.
Yeah.
Okay, I'm through. Somebody else can jump in.
if there are no questions, then I think I'm done.
I have a quick question. Are there any other like filter feeding animals that also suffer from like intaking microplastics?
I'm not sure, but there definitely are other filter feeding organisms.
pretty much any filter feeding organism is going to uptake microplastics.
But we can get mussels pretty easily since we have tide pools near us in Santa Clara.
Exactly. And mussels are a larger filter feeder that have more potential to retain a larger amount of plastics. But yeah.
So it might even be useful at that point to compare the amount of microplastics taken up by mussels compared to some of the other filter feeders in the environment.
It absolutely would. That would 100% be a next step in our project. Yeah. All right. Is there any other questions for Adler?
No question, but good job.